My name is Samuel Bueno, and I am presenting the cell cycle. So the cell cycle's purpose is to reproduce more cells. In that case, we have mitosis and we have meiosis. These two are two types of cell divisions. In mitosis, we have an outcome of two daughter cells, each containing the same amount and the same type of DNA as the parent cell. On the other hand, in meiosis, we have four daughter cells, each containing half the amount of DNA as the parent cell. Now, on meiosis, for a whole meiotic division to occur, it must go through the cycle twice. Most of our cells go through a mitotic division, but our gametes, which is our sperm and our eggs, go through the meiosis. Now, since it goes through meiosis twice, that means we have meiosis 1 and meiosis 2. But before we get into those, I want to talk about the cell cycle. So, in the cell cycle, we have interphase, which is the largest portion of the cell cycle, and then we have M phase. In interphase, we have G1, S, G2. In G1, the cell is in a growing state. It is bringing in nutrients, it is storing energy, it is getting enough for it to reproduce completely and successfully. Once the cell gets a signal, then it is allowed to pass the checkpoint and go into S phase. In S phase, the cell begins to replicate its DNA. After the whole DNA has been replicated identically, it then moves over to G2. G2 is a continuous growing of the cell, and it produces an organelle called microtubules. Microtubules are meant for the M phase. So there's another checkpoint before going into the M phase. And this checkpoint is to make sure that the cell does not contain any toxins, that the DNA has not been mutated, and then it has a sufficient amount of energy to go into the M phase. So in M phase, we have 5 sections. This includes prophase, which is a decomposition of the nuclear envelope and most of its organelles, as well as the centrosomes, begin to move over to the poles of the cell. Once we reach prometaphase, that is when the microtubules make spindles, and these spindles attach to the chromatid, to the sister chromatid, and start pulling them towards the center. In metaphase, all the sister chromatid must be aligned in the middle of the cell, and we reach another checkpoint. Now, this checkpoint is for the cell to make sure that each sister chromatid has a microtubule spindle connected to its centromere. If there is a sister chromatid that has not been connected, the cell does not move forward and dies. In the scenario where it does, we go into anaphase. Now, anaphase is these microtubules start pulling back their spindles and start pulling off the sister chromatids from the centromere. This divides the chromosome into two. That's why we have two sister chromatids. And in telophase, all the sister chromatids that have been pulled apart reach the polar ends of the cell, as well as the nuclear envelope starts forming again. We have all the organelles to begin to reform, and we also have a cleavage. A cleavage furrow is as the cells begin to divide, there is a furrow between these cells, and once they are far enough from each other, it divides. In plants, however, they don't have a furrow. They start forming a plate in between both cells that once it's fully made, it just divides the cells into two. After telophase, right before the division, we have cytokinesis. Now, cytokinesis is not part of the M phase. It's its own little section. Cytokinesis is as the actual division of the cell. Cyto meaning cytoplasm. Now that we have this information, we can move over to meiosis 1 and meiosis 2. So the difference between meiosis and mitosis, aside from the outcome, is that during prophase 1 of meiosis 1, we have the chromosomes of the first parent that pair up with the chromosome of the homologous chromosome of the second parent, and as they pair up, this creates crossing over. During crossing over, both parent cells interchange different genes, this leading to a mix of that DNA strand. And then once that happens, the cell keeps going through all the M phase, all the other four M phases. Once the two cells have been divided completely, it goes through meiosis again. Therefore, we get four different cells, well, four daughter cells. And after prophase one of meiosis one, everything, all the other steps are the same. In meiosis, the cell has to go through interphase all over again, and it has to go to cytokinesis and all the five M phase steps. Thank you.